Great. Hello, everyone. I'm excited to talk about our work examining prenatal polydrug exposure and effects of timing on neonatal brain connectivity. Prenatal polydrug exposure, or PDE, has widespread effects on developing brain networks, particularly in higher order networks associated with reward processing, emotion regulation, and social cognition. PDE is also related to behavioral and cognitive effects that emerge during infancy and persist through childhood and adolescence. Work in animal models has shown that the timing of PDE during pregnancy significantly and differentially impacts offspring outcomes. However, most human fMRI studies collapse PDE across the entire prenatal period when assessing drug exposure impacts on the infant brain. This limits our ability to detect timing effects that are associated with PDE and is a major gap in the field since we know that the fetal brain develops along a discrete timeline such that exposures during different sensitive periods in prenatal development could have different impacts on postnatal neurobehavioral outcomes. Here we examine how the timing of PDE impacts brain functional connectivity at birth and whether these timing differences are related to later behavioral outcomes. To do this, we leveraged two data sets collected in infants with and without drug exposure. The first data set was collected between 2016 to 2020, and the second was collected from 2008 to 2014. In each data set, we had a group of drug-free controls and a group of infants with prenatal exposure to any combination of substances, including alcohol, nicotine, marijuana, cocaine, opioids, stimulants, depressants, and other substances. Information on drug use during pregnancy was collected using the timeline follow-back interview. Using this data, we then separated infants with PDE into two subgroups based on whether infants were exposed across all 3 trimesters, the PDE-F group, or whether infants were exposed only in the first and/or second trimester, the PDE-T1-T2 group. We didn't have enough N to be able to look at discrete trimester exposure, so that's why we separated into PDE-F and PDE-T1-T2. The percentage of subjects in each timing subgroup who were exposed to different substances varied between the data sets. There was a lot of heterogeneity in terms of the polydrug profiles that infants were exposed to, but for both data sets, a high percentage of T1, T2 subjects were prenatally exposed to nicotine and alcohol. Polydrug exposure is the norm rather than the exception in this population, and infants in our sample were prenatally exposed to up to as many as 7 different substances. So in these graphs on the y-axis, I'm plotting the average number of unique substances that the groups were exposed to, with the PDE-F group in red and the PDE-T1-T2 group in blue. And you can see that the T1-T2 infants were exposed to fewer substances on average compared with the full group. In terms of data collection, resting state fMRI scans were collected at 2 weeks of age, and infant behavior was assessed by the Bayley scales of infant and toddler development for cognitive, language, and motor development. In dataset 1, the Bayley was collected at 3, 6, and 12 months, and in dataset 2, the Bayley was collected only at the 3-month time point. For both of our datasets, the groups were matched on sex, race, gestational age at birth, age at scan, and motion. We took a data-driven approach to determine differences in functional connectivity. First, we conducted a whole brain heat map analysis to assess pairwise differences in functional connectivity between subgroups. To do this, we extracted the time series from 223 neonatal functional parcellation regions of interest, or ROIs. Then we calculated the functional connectivity between each ROI to the whole brain. And next, we ran a functional parcellation level linear regression, covariating for birth weight, maternal education, and maternal depression. Then we generate a heat map showing the percentage of significantly different connections. We thresholded these heat maps using p-value thresholding at p less than 0.05, as well as thresholding with bias-corrected Hedges G effect size to avoid sample size biases among the subgroups that we analyzed. Next, we assessed the degree of overlap between the two datasets to see whether the main findings would replicate. For each ROI's connectivity map, we calculated a percent overlap by taking the number of significant connections that were shared between the two datasets and divided that by the total number of significant connections across the two datasets. Lastly, we ran a canonical correlation analysis, or CCA, to examine whether differences in neonatal connectivity were related to later behavioral outcomes. To do this, we took the functional parcellation heat maps showing the timing subgroup differences between PDE-F and PDE-T1-T2 from step 1, and we back-projected those maps to the single subject space to calculate within-network connectivity measures for each subject. We used Bailey outcomes on cognitive, language, and motor development, and we regressed out the same covariates of no interest, birth weight, maternal education, and maternal depression from these data. We then ran CCA to find the maximal correlation between the brain data and the behavioral data, and ran permutation testing across 100,000 randomized permutations to identify significant CCA modes that reflected a significant relationship between brain and behavior variables. Lastly, we ran an exploratory post-hoc analysis looking at between-network connectivity and whether that may relate to behavior. All right, first looking at the whole brain heat map results in dataset 1, PDE infants showed altered functional connectivity, particularly in frontal and sensory motor regions. Direct comparisons of the timing subgroups with control infants revealed different but overlapping topology of connectivity differences. And when we directly compare the two timing subgroups, we found widespread differences in connectivity across frontal, temporal, and parietal areas, suggesting that brain connectivity is differentially affected by the timing of PDE. I'd like to note here that despite the difference in sample size between the timing subgroups, we observed a similar degree of alterations when comparing each PDE timing subgroup with control based on this p-value thresholding. When we directly threshold based on the bias-corrected Hedges G effect size, which accounts for the sample sizes, we found that differences in the T1-T2 group revealed much larger effect sizes. Looking at dataset 2, we found a similar pattern of results, with differences in T1-T2 again associated with larger effect sizes. We next assessed the amount of overlap across the two datasets, and looking here at the p-value thresholded heat maps, overlap of the results was concentrated in frontal and temporal areas. Looking at the effect size thresholded heat maps, we found that around 8% of the connections showing the largest timing-related effects replicated between the two datasets. Next, looking at the brain behavior associations, our CCA identified a significant relationship between neonatal brain connectivity and 12-month Bailey scores in dataset 1. And here I'm showing the plot of the principal CCA mode with the brain canonical variables on the x-axis and the behavior canonical variables on the y-axis. Looking at the plots of the canonical coefficients for each model parameter, we found that within network connectivity in the subcortical, default mode, and sensory motor networks were the best predictors of 12-month Bailey scores. 12-month Bailey language scores were best predicted by the neonatal connectivity measures. Interestingly, lower order networks were positively related to language and motor scores, and connectivity of higher order networks was positively related to cognitive scores, indicating that a balance between lower versus higher order network development may set the stage for later behavioral outcomes. Lastly, in an exploratory post-hoc analysis, we found that between network connectivity with the subcortical network was the best predictor of 12-month Bayley scores. This suggests that the subcortical network may serve as an important hub during early development as the neonatal brain sets up functional connectome. To close out with some takeaways, at birth, the timing of PDE is associated with distinct effects on the brain. Even though the T1, T2 group experienced a shorter duration and lower degree of exposure, early drug exposure during the first and second trimesters had the greatest impact on neonatal functional connectivity. This points toward potential interactions between the differential timing of PDE and specific neurodevelopmental processes during the prenatal period. Lastly, our CCA showed that timing-related differences in functional connectivity at birth are related to 12-month behavioral outcomes. This indicates that these early differences may alter neurobehavioral trajectories over the first year of life. Taken together, our results emphasize the importance of examining the timing of environmental mediators on neurodevelopment. This provides a unique opportunity not just to identify risk, but also resilience and plasticity, so we can identify what might be protective, since both are equally important in informing early interventions. And finally, multimodal integration of brain-based measures, pre- and postnatal environmental factors, and later behavioral outcome is crucial for understanding mechanisms that drive altered developmental trajectories. With that, I'd like to thank the members in the lab, our collaborators, and our funding sources. Thank you for your attention, and please stop by my poster after lunch to say hi, and I'm happy to take any questions.
Excellent talk. So we have time for a question or two. So I'm going to combine two of these questions into one. So how did you account for heterogeneity in PDEs? So for instance, you consider antidepressants and drugs like cocaine in the same manner in the analyses. So you just talk a little bit about the heterogeneity issue.
Yeah, that's a good question and something we're constantly running up against when studying prenatal drug exposure. So as I mentioned in the talk, polydrug exposure is the norm, and we try our best to be able to match wherever possible. It's just not always possible. I know that's not a great answer to this question, but another direction that we're hoping to go in is to look at dosage in addition to timing, because just as people have collapsed across timing, people also collapse across different dosages. And I think future work in like the HBCD data set, where we have just a higher N, we'll be able to start parsing through that heterogeneity.